Hello, all. Welcome to this session of life processes. Hope you are doing affirmations before you begin this any session of mine. If you remember the affirmation, I am learning every day by chunking and I am improving by each passing day. So let's begin today's lesson. This is a very simple. This is about all the activities that we have in NCERT in plant nutrition part. But before we begin, let's do a quick recap of what we studied in last class. In nutrition, we have two different types of nutrition, autotrophic and heterotrophic nutrition. And we discussed autotrophic nutrition. What is autotrophic nutrition? A kind of nutrition where organisms prepare their own food, whereas heterotrophic nutrition is a kind of nutrition where organisms cannot make their own food. So this is we studied there in heterotrophic nutrition. Again, we have different types of nutrition. We will discuss those different types when we study nutrition in animals. Let's move ahead today. This is an activity which is given in NCERT. There are two activities in nutrition in plants. As you remember, we had four requirements for photosynthesis. One was the carbon dioxide, the other was the water, the third one is light, and the fourth is chlorophyll. So, how do we prove that these are required for plant nutrition? This is there are two of them are given in your book. One is chlorophyll is necessary for photosynthesis, and the other is carbon dioxide is necessary for photosynthesis. Based on these two activities, we can formulate the third one also. Light is necessary for photosynthesis, and water is very obvious. If you do not water plants, the plants are going to die. So that is necessary for photosynthesis. Let us take activity 1. What does it say in activity 1? We have to take a potted plant with variegated leaves. Variegated leaves, what does it mean? A leaf having two different colors. One has to be green, the other can be any other color. You might have seen corotan plants which has yellow parts in it. There are some plants which have white parts in it. There are plants with red color also over there. So we have to take a variegated leaf which has green color, as along with that it has some other color. Then we keep the plant in dark for 3 days so that all the starch gets used up. Why do we keep the plant in dark for 3 days? So that all the starch which was stored in the plant gets used up. So this says that starch is the storage material of plants, isn't it? So in plants, after photosynthesis, glucose is being prepared, and that glucose is being converted into starch, and that is being utilized by plants for its different activities. So we keep the plant in dark room for 3 days so that no photosynthesis occurs, and whatever starch has been stored in the plant, that gets used up. Now keep the plant again in sunlight for about 6 hours. Why do we do this? So that plant starts photosynthesizing again, and it starts preparing its glucose, and that glucose is again converted into starch. So this is the duration of 6 hours. Now what we do? We take a leaf from the plant and then mark the green areas with the help of a tracing paper. After this, our experiment begins, real experiment in laboratory. We dip the leaf in boiling water for a few minutes, and then after this we immerse it in a beaker containing alcohol. This here we have to take a precaution. Let me show you if I can show it in this page. Let me change the color of the pen first because this is blackboard. Yes. How do we use this alcohol? We take a beaker in which we have water. Water is there in this beaker. and then we take a test tube alcohol is present in this test tube. We put the leaf in this test tube and boil it. We do not directly boil the alcohol because alcohol is highly inflammable. Why are we using this water bath? This arrangement is known as water bath. Why do we use this water bath? Because alcohol is highly inflammable. If we will directly heat the alcohol, what will happen? It may catch fire because it catches fire very easily. So this is we dip the leaf in alcohol and boil it using a water bath. We will boil it until the alcohol begins to boil. What will happen because of this alcohol boiling? The green color of the leaf that we have in the leaf, that is green and bluish, they will start coming out from, not blue, yellow, they will start coming out from this, and this liquid that we had taken as alcohol, that will start turning green slowly and slowly. All the chlorophyll and other pigments of the leaf, they will be dissolved in alcohol, and the color of alcohol will change. So what happens to the color of the leaf? A leaf becomes transparent, white in color. And what will be the color of the solution? It will become greenish in color because all the chlorophyll is dissolved in alcohol. Why does chlorophyll dissolve in alcohol? Chlorophyll is an organic compound. This is also an organic compound. So organic compound dissolves in an organic compound. Now what we will do? We will take out this leaf from this and rinse it in running water, and then so that all the alcohol that is there is removed. Then we will dip the leaf in dilute solution of iodine for few minutes, and then take out the leaf and rinse off the iodine solution also. What will happen in this? The portions, let me change the color once again. The portion which were green in color, that changed into blue black. That changed into blue black, and iodine changes starch. If starch is present over there, the color of that starch is changed as blue black. Okay, this is why this happens. So this is what will happen when we take out the leaf from the iodine solution, we rinse it off, and then observe the change of the color that happened. Then what will happen? Wherever we had the starch, wherever we had starch or wherever green color was present in the leaf, that is chlorophyll was present, there we have starch formation, and that turns into blue black because iodine turns starch blue black. So this is one activity which shows chlorophyll is necessary for photosynthesis. What precautions we have to take? We have to immediately observe the results because after some time the starch will start being distributed to different parts of the plant. So that is why in the beginning we had removed the starch by keeping the plant for 72 hours in dark room, because starch was already there in the plant, it was distributed all over the plant. And then we will do the experiment immediately after 6 hours so that starch does not start getting distributed. These are the precautions that we have to take. Next, few questions that are there is, why do we keep the plant in dark for 3 days? to why do we boil leaf in alcohol using water bath? Third, what is the test for starch? Is, is it necessary to boil the leaf in alcohol? If yes, explain. Okay, why do we keep the plant in dark for 3 days? I hope you can answer this. We kept the plant in dark for 3 days so that all the starch is used up. Why do we boil leaf in alcohol using water bath? So that alcohol is highly inflammable so that it does not catch fire. What is the test for starch? Iodine solution turns starch blue-black. Is it necessary to boil the leaf in alcohol? Yes, because alcohol removes all the colored pigment from the leaf, and if colored pigments are removed, it is easy to get the results from the experiment. When we put iodine solution, the green color of the chlorophyll will not interact with the blue-black color, and we would be able to justify the result more easily or get the result more easily. Now the second activity is take two healthy potted plants. That is, we take two pots which are having plants in it and nearly of the same size so that we can compare it more easily. We the same experiment, first step will be same for in each of the experiments. We keep it in dark for 72 hours so that all the starch is used up, and then we place each plant, cover it with a what do we call it, bell jar made up of glass. But before covering it with bell jar, we in one of the near one of the potted plant, we put a watch glass which contains freshly prepared potassium hydroxide. Okay, what is the use of this potassium hydroxide? Potassium hydroxide is capable of absorbing sunlight from the atmosphere. Now we cover both the plants with separate bell jars, which is made up of glass. Why made up of glass? So that light can penetrate through it. After this, we seal the bottom of the bell jar with the help of Vaseline so that no air can enter inside. We make it airtight. No air is getting inside from outside. Now we keep the plant in sunlight for 2 hours so that photosynthesis begins and new starch is being formed. After this, what we will do, we will pluck leaf from both the plants and check for the presence of starch as we did in the last experiment. What did we do? We will take the leaf, we will boil it in alcohol, then we will put it in iodine solution, rinse off the excess iodine, and then we will see what is the result. What will be the result? As I told you, KOH, freshly prepared, sorry, KOH is able to absorb carbon dioxide. So in the jar where we have KOH, in this jar what will happen? This area which was covered, this portion of the bell jar which was having some air in it, from that all the CO2 has been absorbed. So plant is not getting any CO2, so no photosynthesis will take place in this jar. No photosynthesis takes place in this jar because all the CO2 which was present in this jar, we have made it airtight, so nothing can come from outside. So which was already present in this jar was absorbed by this QH solution. This is not having any QH solution, so whatever amount of CO2 is present in these areas, they are utilized by the plants for photosynthesis. So CO2 present in these areas are utilized by the plant for photosynthesis. So what will be the result if we take it from, let me change the color of, if we take it, the leaf taken from jar A will show no color change because no photosynthesis has taken place. As all the CO2 was absorbed, no photosynthesis took place. So no photosynthesis means no starch. So no color changes there when we test it with iodine. In the second one, what will happen? The leaf will turn into blue-black. That means starch is present there. From where did that starch come up? By photosynthesis. So carbon dioxide is necessary for photosynthesis. Here we have removed all the carbon dioxide in this one. So what happened? No photosynthesis took place in this jar. So we can say carbon dioxide is necessary for photosynthesis. Let me move to next page. So few questions here. What is the use of KOH solution? Why do we make the experimental setup airtight? Why should we take freshly prepared KOH solution? So what are the answers? What is the use of KOH solution? KOH solution is used for absorbing CO2 from the experimental setup. Why do we make the experimental setup airtight? So that no air enters inside and the plant does not get any carbon dioxide from outside source. Why should we take freshly prepared KOH solution? This freshly prepared KOH solution gives better results. Okay, and it has to be a solution. We cannot take it in solid form. So these are few questions that comes from this part of the chapter. Now the third one is light is also necessary for photosynthesis, isn't it? Light is necessary for photosynthesis. The same experiment we can perform in a different way to show that light is necessary for photosynthesis. We take a potted plant, keep it in dark for 72 hours. Take a potted plant, let me write it so that you remember. Keep it in dark for 72 hours. What happened? All the starch has been used up. Now again keep it in sunlight for 6 hours. What will happen? Photosynthesis begins. Starch has formed. But before keeping it in sunlight, because we are showing this light is necessary for photosynthesis, we have one more step over here, that is, this will become number 4 then. What we do here, before putting it in sunlight, we cover a part of leaf from both sides with the help of opaque paper. Why opaque? This is the step that we do before keeping it in sunlight. Now, how do we cover it? Let me draw a leaf. Suppose we have a leaf over here. We will cover this leaf This is opaque paper by which this leaf is covered on both the sides so that no air light can penetrate into it. So let me mark these areas A, B and C. Let us see what will happen. So this is our leaf. After keeping the plant in light for 6 hours, we will pluck that leaf. This is A, this is B, this is C. We will remove the strip that we had put over there and then test it, boil it in alcohol using water bath, put it in iodine solution and then rinse off excess iodine and see the result. What will be the result? A will, because it was not covered, this will show blue-black color. That means starch is present over there. C will also show blue-black color. But B, there is no change in color because no photosynthesis took place in this. Why no photosynthesis? Because this portion was covered with an opaque paper. An opaque paper did not allow light to pass through it. Opaque objects do not allow light to pass through it. So light could not reach this part of the leaf. Though it was kept in sunlight, light could not reach this part of the leaf. So no change in color, no photosynthesis took place in this part of leaf. So this shows that light is necessary for photosynthesis. Light is necessary for photosynthesis. So today we learned CO2 is necessary for photosynthesis, light is necessary for photosynthesis, chlorophyll is necessary for photosynthesis. These three, CO2, light and chlorophyll. We had activity for these, and water, we can observe this. If you do not water plant for a longer duration, plants die. So all these four are necessary for photosynthesis. So this is all about nutrition in plants. Tomorrow I will come up with some questions from this part of the chapter. So revise it if you have not done it, and then we will discuss some questions tomorrow. Thank you for watching this video. Enjoy learning. Subscribe the channel to get the notification, like it and share it. Thank you.